I'm Dan Benjamin, for those of you who haven't met me yet. I'm one of the co-organizers of this Summer Institute, along with David Cesarini, who's sitting in the back. We are very excited about the course that you're going to go through over the next couple of weeks. I think we've put together a great set of instructors. I'll tell you a little bit about them. Putting together the course has required us to think through a lot of to try to structure our own way of thinking about social science genomics and put it down on slides and in problem sets. And so we're very excited to share that the product with you. But you're guinea pigs. This is the first time we've done this, so we apologize for any of the hiccups we have along the way, such as the repeated corrections to the first problem set. Please bear with us, and please do give us feedback. We will probably be doing this again, and we want to know how to do it better going forward. So feel free to talk to us anytime, any way, by email, by coming up to us, anonymous tip, anything you're comfortable with. We are extremely grateful to a number of different groups and individuals for helping to make this possible. The Russell Sage Foundation provided funding and support. Liana Shatrath from the Russell Sage Foundation arranged our contract with the Rutgers University Inn and Conference Center, and we're also grateful to this conference center. My wife, Samantha Cherney, raise your hand, Samantha, helped with dozens of logistical issues that have come up along the way. Ad Health, as you'll see, was willing to provide data to us for your benefit, to use that data actually before they've made it publicly available. They literally just cleaned the genetic data and then said, now we'll give you access to it. So they've been incredibly helpful. The National Bureau of Economic Research is where we at the Social Science Genetic Association Consortium host some of our powerful computers that allow us to do genomic analysis. We've basically dedicated those computers for these 2 weeks to you, so you have access to those cores for analyzing the Ad Health data, and we're grateful to the NBER for supporting that. And to the instructors, all of whom are completely volunteer. They're just giving their time because they, like us, think this is a really important program to conduct. So Stephen Glauser from the Russell Sage Foundation is here, and I thought maybe he could say a thing or two about some of the other things the Russell Sage Foundation offers in terms of grants and visiting fellowships that you might be interested in.
Thanks, Dan. So I'm from the Russell Sage Foundation. We're sometimes called a think tank, sometimes called like a research institute. We fund a lot of social science research in our main program areas. Those include social inequality, the future of work, race, immigration, and ethnicity, and I'm missing one. Oh, behavioral economics. And so this kind of project is a very interdisciplinary thing, which is something that the Russell Sage Foundation has really concentrated on for the past, the better part of 3 decades, since it was one of the beginning funders in behavioral economics, which is really a combination of psychology and economics. And so here we're hoping to bring in social science with biology and genomics, and we're hoping to kind of help spur the same kind of growth of a field that we feel is being quite exciting. The methods that we do funding are, we have, in those program areas, we have regular funding round 3 times a year. We're actually expecting to have an RFP specifically for biology and the social sciences that we expect to come out later this summer or early fall. We're not really sure on the timing yet. But that's something that'll go across all of those program areas, because we'll recognize that these things affect things like race, immigration, and ethnicity, and like social inequality, and so those can go across the entire bandwidth. And so we're really interested in interdisciplinary projects. So look for that RFP forthcoming later this summer or fall, like I said. And you can find out more information about all those program areas at russellsage.org, or you can come up and ask me whenever we have free time later. All right, thank you.
Thanks, Stephen. So I should also say, throughout this talk, and actually all of the talks in the Summer Institute, I'm an economist, but even for the non-economist speakers, we're all used to being interrupted with questions as we speak. And since there's no specific time set aside for question and answer for these presentations, you should please raise your hand. Actually, that's better than directly interrupting. Raise your hand if you have a question or a comment, and during the talk. Okay. So this is the Summer Institute in Social Science Genomics. What is social science genomics? Well, it's the use of genomic data in social science research. And we think that there's a lot of possible potential payoffs that are likely to come out of incorporating genomic data into the social sciences. And actually, most of the lectures today are going to be about that, about those payoffs. At the same time, there are many challenges to actually doing that, to doing credible research with genomic data, interpreting the results of that research, and then communicating those results to the public, to the media. Tonight's lecture, the dinner lecture, is going to be about some of these ethical issues, which I think is, these kinds of ethical issues and communication issues come up in all research, but I think that they're especially important for us to think about in social science genomics because there's a lot of baggage, historical baggage that this field carries around, and we need to be aware of that whenever we're thinking about and talking about this kind of work. Much of the content of the program itself, the lectures, are going to be about how to do good, responsible research that overcomes the research challenges to doing the work. So why does the Russell Sage Foundation, why are we interested in having this Summer Institute now as opposed to a few years ago or waiting a few years from now? Well, in the last few years, there have been very rapid advances in our ability to measure genetic variation across people, both on a greater and greater scale, moving from 10 years ago, just measuring a few genetic variants at a time, to more recently doing whole genome association or genome-wide chips that measure hundreds of thousands and now typically 2.5 million, moving in the near future to genome sequencing. And you'll hear more about this later today. So that's an increase in scale and also falling costs holding fixed the technology for doing the genotyping. This is a figure that NIH put out that I think what they've done is looked at basically the cheapest per megabase technology for doing genotyping, and they've graphed over time what that cost is. This line shows Moore's law, which you may know is the rate of increase of processing power for computers over time, which is considered to be an extraordinarily benchmark for extraordinarily rapid technological progress. And you can see that genotyping costs have actually been falling at a faster rate than Moore's law. In addition to, as a result of these falling costs, the data are now increasingly available in data sets that we would be interested in as social scientists. A couple of years ago, the Health and Retirement Study made genome-wide data available. AdHealth now has genome-wide data available. The UK Biobank last year made a huge amount of data available on a very large number of people. And more social science data sets or data sets with relevant social science information are becoming available. That creates the opportunity to use that data to do social science genomics research. And there have also been, and I think this is especially relevant for the question of why we didn't have this Summer Institute earlier, it's really only in the last couple of years that there have been dramatic advances in our understanding of which specific genetic variants are associated with behavioral traits, and our ability to aggregate that information into predictive scores that have enough predictive power to be useful in social science research. Not only have there been dramatic advances, but it's pretty clear that the rate of increase is extremely rapid and convex, that we're actually at a point now where in the next couple of years, the amount of predictive power is going to dramatically increase relative to where it is today. So I think really now, right now, the time is ripe for bringing genomics in a pretty big way into social science research. Okay, so who are we, the instructors? Well, we represent a range of different disciplines in the social sciences: economics, sociology, psychology, political science, genetics, bioethics. I think it's fair to say that all of the instructors that you'll see are among the leaders in this area. Some of us are members of the Russell Sage Foundation Biology and Social Science Working Group. So that's myself, Dalton Conley, Coulter Mitchell. Most of us are also core members or very close collaborators with the Social Science Genetic Association Consortium. And you'll hear more about the consortium later this week, but I'll just say now that the SSGAC coordinates large-scale genome-wide association studies for behavioral phenotypes. A lot of the recent advances in connecting genomic data with behavioral traits is a result of the work of the SSGAC. The goals are to create research tools that researchers can use in their own research, like polygenic scores that have predictive power for behavioral traits, to promote good research practices, like doing ex-ante power calculations, pre-registering and completely reporting all methods in the papers, publicly posting results. And you'll see these are themes that are going to come up in the course of the Summer Institute as well, and a commitment to an accurate communication and interpretation of the findings of the research. As I mentioned before, the two organizers of the Summer Institute are myself and David Cesarini. We are co-directors of the Social Science Genetic Association Consortium together with Philip Kohlinger. Maybe I should say something about my own story of how I got involved in doing social science genomics research. And maybe David can tell you some of his story of getting involved when he speaks tomorrow. I've actually been interested in this area for a very long time, since I started graduate school, which was in 2001. At that time, the Human Genome Project had just been finished, and neuroeconomics, which is this field of trying to understand how the brain implements decision making, had just started as an area. And I was at a conference with some of my advisors in graduate school, and we heard a presentation on neuroeconomics, and it was like the craziest thing. Like some economist was studying the brain, and we were talking about it afterwards and thinking, well, that's so crazy. What would be the even crazier thing? Well, to go to genes. And so, that seemed like it would have to be exciting someday. And so I started thinking about it and trying to learn about, trying to get access to data, which was impossible at that time. In 2004, I finally got access to some data with my advisors, which was Icelandic data. And we did what was called a candidate gene study, which was totally standard at that time. It took, I ended up taking a bunch of trips to Iceland as a graduate student, which was awesome. But then, in the end, we found some results that didn't replicate, which is kind of a typical story, and actually in a lot of candidate gene research, as we'll talk about later this week. That was by around 2009. And so at that point, my collaborators and I started to think a lot more about what was going wrong. Why were our results not replicating? What would be a research strategy that would actually potentially work in this area? And we looked to medical genetics research, which had made a transition from the kind of candidate gene work that was standard there too, into genome-wide association studies with much, much larger samples in around 2006. And so with David, we came to the realization that that would be potentially a productive way to make progress, and got in touch with Phil Kohlinger, who had been pursuing a consortium effort for entrepreneurship independently. And we kind of made that much broader, made it into social science. And then since we've been working on the SSGAC, we found that what worked in medical genetics also seems to be working in social science, and we think we understand now the good reasons why that's the case. So it's been a very, very long path for me, with a lot of frustration along the way, but I think that we're now at a point where we're starting to see real progress. Okay, so that's us, or me. Who are you? We had a very competitive application process. We got 60 applications and selected 26 participants. We wanted to keep the Summer Institute relatively small so that it would feel more intimate, and so that we, the instructors, could get to know each of you. But we also are videotaping, as you know. We're videotaping the lectures, and we'll make the videos and the problem sets publicly available so that anyone who's interested in the material can follow the course afterwards. You guys represent a wide range of fields as well. And we selected you because we believe, based on the materials you submitted, that you are likely to be a future leader in your discipline, and to have a lot of impact on your colleagues and your students. And so you are a special group, and I think you'll learn a lot from each other. In fact, you'll probably learn more from each other than you do from us, which is the part of the goal. And we're looking forward to learning. A lot of you are expert in specific areas of this kind of research, and we're looking forward to learning from you too over the next 2 weeks. Okay, so what are the goals of the Summer Institute? Well, one of them, probably the most important, is to help you, to spur you to develop your own research ideas that will be high quality, good research in this area. We're also hoping that you'll learn how to implement and interpret new methods that you may not have seen before. We'd also think that you'll become a more discerning consumer of this kind of research. So even if there are topics that we cover that are not things you yourself will do research in, I think you'll be better able to appreciate what are the challenges, what is good work in the area. And that's particularly important when evaluating the work which is claiming to have found genetic associations or estimating a polygenic score, so that you can see, is this a reliable base of knowledge to build on in your own work. We think you'll learn about some of the useful data that's out there and some of the software that's used to analyze it. And another goal is to learn about good research practices that apply actually even more broadly to empirical research, not just in social science genomics. Okay, so let me say something about our teaching philosophy, because this is a little bit of a different kind of program than some of the other programs that are out there. So for one thing, we worked hard to make this Summer Institute not just a series of lectures by distinguished people, but a coherent course that kind of builds from beginning to end into a body of knowledge. Because modern genomics research is ultimately empirically driven, we thought it was important to give you access to real data. So that's important too, because often in these kinds of learning experiences, because genetic data has all sorts of IRB and privacy restrictions, what students are given is access to simulated data. But we wanted to give you actual AdHealth data, so we, and particularly Mark, worked hard to make the logistics for this work out, which were not easy. I mean, we needed to get all sorts of permission from AdHealth and from the NBER and deal with IRBs. And so you don't have to jump through any of those hoops other than signing a few forms to get access to these data for 2 weeks. And also the hardware and software and computing power that you'll need in order to analyze the genomic data on the NBER server. Now, another distinction with, I think, a lot of the other instruction in this area is that a lot of it is about just learning how to run code. And in our view, the core theoretical ideas are at least as important. So understanding conceptually what's going on is important for identifying what are the good research questions and thinking about the right interpretation of the research findings. And so there's a lot of emphasis in these next 2 weeks on the key theoretical ideas, which we're going to try to convey through the simplest models that we can come up with to convey those ideas. And then as a result of this emphasis on key theoretical ideas through simple models, the problem sets are really central to the program. As I think you've already seen with problem set 1, they're part of the teaching. We've done the best we can, or we will continue to do the best we can, to have uniform notation so that you can go between the problem sets and the lectures, and it will all fit together in a common framework. That's always very hard to do, and I'm sure we're going to fail to some extent, but we're going to do our best to make it all a coherent instruction. Okay, so this is going to be an intense, immersive experience for you, and for us too. We the instructors, we've put all of our research and other life commitments on hold for 2 weeks to dedicate it to this program. For you, all day is going to be filled with lectures, and then there are going to be these problem sets that are going to be happening all the time. So you'll have the evenings to do those, and we also want you to be meeting each other, brainstorming research ideas. So it'll be intense. We encourage you to go to office hours. We'll talk a little bit more about how those will work, but that's partly to talk about the problem sets, which are and the lectures, but also to talk about research ideas. So take advantage of that, especially for the faculty who happen to be visiting in your area. Work on the problem sets in groups. Part of the criteria for admitting you to the Summer Institute was that you had a strong quantitative background, but some of you have stronger backgrounds than others, and some of you will be more familiar with some of this material from previous work you've done. And I think I think it's important for those of you who understand things relatively better to help those who are struggling more, and brainstorm with each other about possible research. The problem sets, as I mentioned, the purpose is to teach the material. I want to be clear that they're intended to be challenging. So, I know a number of you have found the first problem set to be challenging. That's good. I mean, the difficult thing about the first problem set is that you were on your own to do it. Going forward, we're hoping you'll work on them together in groups and help each other, discuss them with the instructors. And one thing I want to emphasize is that it's tempting, or sometimes one falls into the habit of doing a problem set and turning it in and then basically forgetting about it. You get the solutions, but don't bother to look at them, or only look at the parts that you struggled with. I encourage you to go through the solutions for the entire thing, including the ones that you think you got right, because that's where you learn, when you see the right way, the right answer compared to what you thought, or working through the problem sets, I think, will be very instructional. I should also say that two of the instructors, Patrick Turley and Mark Fontana, have very kindly volunteered to be essentially TAs for the problem sets. Patrick will be getting here later today, and he's particularly he's kind of going to specialize in giving help on the theoretical parts of the problem sets, and then Mark, who many of you have already met, will kind of specialize in giving help on the data computing aspects of the problem sets. So let me actually say more about the data computing part of the problem sets now. This will be problem set 5, which is going to be, although we're calling it problem set 5, we're actually going to hand it out today, so that you'll have as much time as you want to work on it throughout the next 2 weeks. It's going to be focused on the software, using the software and analyzing the real data from AddHealth. Mark has put together a set of PowerPoint modules that teach what you need to know in order to do the problem set 5, so you can work through it at your own pace, and of course talk to Mark as you want help. An ideal time to work on parts of the problem set is when in lectures we're covering the theory related to the data analysis. But of course, you can do it at whatever pace you want. And part of the point of doing it this way, of giving you access now and having you work through it on your own, is that once you've done that, you can use the data. You'll then know how to use the software to begin exploring it on your own for research questions. So you can do that starting later today, and start looking in, running some regressions that might lead into a research project. Mark, do you want to say anything more?
Yeah, sure. You just kind of stand here and talk. So
No, you should come up here, because we have a microphone goes into the.
I have a few notes, but you managed to cover a lot of it. So yeah, with regard to the server, some of you have already received passwords from me for your login information. If you haven't yet, I urge you to come to me individually at your earliest convenience after the lecture, so I can give you your password. That way you can get access to the server. I'll also be distributing on this thumb drive code books, which give you kind of a summary of all the data that are available, mainly the phenotypic data, which is extremely extensive. The code books are enormous. It'll be a really good, I think, opportunity to potentially brainstorm ideas, just taking a look at them. The other thing is, if there are specific data sets, summary data that you want loaded on the server that you'd like to use, let me know. I am happy to entertain reasonable requests to put data that you can use on the server that everyone can access. The other really important thing is, as Dan said, a lot of work went into getting these data in place. And one of the big things that is highlighted in a lot of the forms that you signed is, under no circumstances can you download any of the data. Please don't do that. That will get everyone in trouble, and we can't do something like this again if that happens. But I don't foresee that being a problem. Everyone works on the server. If you haven't used Linux before, it'll be a learning experience. These modules I've created will hopefully lead you to that experience. I'll also be explaining the first module, specifically lecturing on it, I think, later today. But if you have any questions throughout the experience, that's what I'm here for. So I'll go ahead and pass out the problem sets and whatnot.
Well, let's wait till after the lectures today.
Okay, beautiful. Cool.
Okay. So this is actually repeats a little bit of what Mark said. So Mark can provide you, if you haven't gotten it yet, he can give you your password, but only in person and only on paper. Do not put it in an electronic form. And as Mark already emphasized, don't download anything from the server. And the forms you've signed say that you are responsible for data security and respondent privacy. So be responsible interacting with the server. OK, so office hours are going to be available for any of the faculty who are here at the time when the office hours are scheduled in the program. Several of the instructors are going to be around for all 2 weeks. So David and me, Mark, and then Patrick will be arriving later today and staying throughout. And we'll announce the office hours. The office hours for later today we'll announce later today, and we'll announce them as they come up. OK, so I want to say a little bit about an outline. Actually, most of the rest of the time I'm going to spend kind of guiding you through what the next 2 weeks are going to be like in terms of material. And then I'll say a little bit about the uses of genetic data in economics research. So day one today is all about the big picture. So we're going to be talking about the potential payoffs of genomic data for economics, sociology, political science, and psychology. And the purpose of this is to motivate the tools that you're going to be learning throughout the camp, and helpfully get you to think about what you might be able to do in your own discipline that might be exciting, and spur your imagination for potential research topics. And then tonight, we're going to talk about another big picture topic, which is the ethical issues. As I've said, I think it's always important, but especially important in social science genomics, where not only is there historical misuse of applying results of the data, but also an especially high risk of misinterpretation of findings because of the kind of beliefs that that the typical person comes into when thinking about what social science and behavior means, and heightened IRB and privacy issues for dealing with genomic data. OK, so that's day one. Day two is going to be focused on behavior genetics, but we'll also, in the course of learning behavior genetics, we're also going to learn about the benchmark causal model, which you will see introduced in problem 1 of problem set 2. It's these variables are demeaned. This is a purely additive model of where genotype scores are affecting the phenotype. This builds up from the single locus model that you explored in problem set 1, and we'll see those connections in problem set 2. There's a number of assumptions built into this benchmark model. No dominance, meaning that these effects are all linear. No epistasis, meaning that the loci don't interact with each other in affecting the outcome. There's an assumption of Hardy-Weinberg equilibrium, meaning that which rules out correlation, gene-gene correlation across loci, and also rules out assortative mating, which is not a kind of non-random mating, like maybe more highly educated people tend to marry other more highly educated people. It rules out gene-environment correlation, as you saw in problem set 1. It also rules out gene-environment interaction. And it is by assuming that this is the causal model, we're saying that you don't need to worry about any covariates. For example, different ethnic groups in the population that might otherwise confound the relationship. You could just estimate this model and get the right answer. Is going to be important to generalize from that when we move to data. So actually, throughout the course, we're going to be relaxing all of these assumptions. But for understanding many of the core ideas, this benchmark model is going to be extremely useful. Also on day 2, we're going to, in the context of that benchmark model and some of the extensions, explore behavior genetics. So the way to think about behavior genetics is that we're going to take all of these effects of genetic loci and consider them as a single object, which we can call AI, the effect of genetic factors as a whole. And then the main goal in behavior genetics research will be to estimate what's called heritability, which is the variance explained by the genetic factors as a whole, the variance in AI divided by the total phenotypic variance. And talking about behavior genetics research is going to be useful, partly actually because a lot of research in social science genomics is behavior genetics. There are a lot of twin studies and adoption studies being done. It's an important active area of research. But it's also going to be useful when we turn to genomics and talk about the genetic data, because the very same framework and same issues of interpretation are going to be relevant for talking through genomics. The estimates that we get from behavior genetics research, from looking at twins and adoption studies, are going to be informative with regard to doing research with genomic data. For example, the estimates of heritability are going to give us some guidance about the maximum predictive power we could get from a polygenic score that aggregates across genetic loci. And it's also going to be useful because there are methods being developed that have been developed and new ones coming out that, that aim to estimate heritability using the genomic data itself. And it will be useful to have the behavior genetics estimates as a benchmark and also the set of assumptions as a benchmark. So you can see what are the different assumptions that you need to make to do it from genomic data as opposed to from correlations across relatives. Okay. Also on day 2, we will talk about genetic data and data sets. This will be Coulter-Mitchell's talk. As we've said, I think a driving force of research in genomics and social science genomics is data availability. And so it's important to understand how the genetic data itself is obtained from the research participants, what the data sets are that are available or will soon be available, and some of the advantages and disadvantages of using different data sets in terms of the genetic data and in terms of what kind of other data are included in the data set. Okay, so then day 3 will be molecular genetics, taught by Tonu Escu, who's a bona fide geneticist. And it's important to understand the basic biology of genetics, I think, for a few reasons. One is because it'll help us think through when the modeling assumptions that we need to make are reasonable approximations and when they're not, or when there are exceptions to the assumptions that we're making. It'll help us think about the value of new data that are becoming available very quickly. There's all sorts of data. Used to be you could really only get data on large scale SNP data. Now it's increasingly possible to get access to rare variants, to copy number variants, and other kinds of genetic data. And there'll be more and more going forward. So this day will help us appreciate what you could do with that kind of data and what it means. Well, it's important to learn about the biology to understand what the biological mechanisms are that are ending up affecting the psychological characteristics that ultimately matter for behavioral phenotypes. And finally, it's important to understand what's actually going on mechanistically when the environment has an effect on the genetic mechanisms through gene expression and epigenetics. Okay. Then on day 4, we're going to turn to the question of gene discovery. That is, how do we identify which genetic loci matter for which behavioral phenotypes, and how big are the effects, and how do you estimate polygenic scores? Actually, polygenic scores won't be on day 4, but the individual loci will be day 4. So we start with the, we'll go back to the benchmark model and ask about how we estimate these individual beta hats. It's important to understand the approaches and the challenges to doing this kind of gene discovery to be able to evaluate the reliability of the results that are published. And And also because the same three challenges that we're going to be emphasizing about gene discovery apply to applications of using genomic data in social science. So the issues of power, multiple hypothesis testing, and population stratification. Okay, so then day 5 is going to be, instead of about individual loci, it's going to be about aggregating across many genetic loci. So thinking again about the basic framework, we're going to again go back to considering a variable of all genetic effects taken together. And the point here is that although, as we'll emphasize, the individual genetic loci generally have very small effects on behavioral phenotypes, you can often, sometimes, get substantial predictive power by combining the information across many loci, which helps deal with, for example, the power challenges. So one example of exploiting the aggregate information is constructing a polygenic score. That's a predictor variable. It's essentially an estimate of this AI at the individual level that you construct from genomic data. Another example of exploiting the aggregate information is looking at the genetic correlation across traits. So that's saying you take the vector of betas for one trait, the vector of genetic effects, and look at its correlation with the vector of genetic effects for some other trait and say, does it tend to be the case that when one locus predicts more of cognitive ability, it also tends to predict more of conscientiousness. And then a third example is partitioning. So that's asking the question, are the genetic effects, the beta J's, do they tend to be larger for some types of genetic loci than others? And that's useful for example, for helping to learn about the biological pathways through which the genes matter. Okay, so then day 6 is going to be about the environment. So going back to the model, we're going to look at gene-environment interactions, which is the question of how environmental factors modify the individual locus's genetic effects or modify the effects of all the loci taken together. And Mendelian randomization, which is the idea of using the genomic data to estimate the causal effect of some environmental factor. So we'll talk about some of the challenges of Mendelian randomization and cases when it might work. Okay, so then day 7 will be about structural modeling. So to understand what that means, going back to the basic framework, when we say this is a causal model of the effect of the loci on the outcome, what we're saying is if you intervened at conception and changed an individual's genotype at some locus, what would ultimately be the effect on the behavioral phenotype? But so that's what an economist would call a reduced form causal effect. It's not taking a stand about the mechanism, but it's treating these causal effects as a black box. And what structural models try to do is look inside that black box, estimate the particular causal pathways, and that's especially important if what you're interested in is simulating the effects of policy changes, because then you really do need to understand the mechanisms in order to say how some policy change would matter. The structural models do so in a way that allows for multiple causal pathways, potential imperfect match between the causal factor that actually matters in the world and the thing that you've measured, and the potential for individuals to respond to the changes that they're seeing or to information that they have. For example, they might learn their genetic risk, and then that changes how much they smoke. And so Kevin Tom will be talking about structural models in general and how to use them with genetic data in particular. Okay, and then day 8 is going to be focused on issues of computing. Now, this is important because genomic data sets are large and because some of the analyses are very computationally intensive. And because of that, good research practices for data handling and coding, which matter in all empirical research, become especially important in genomics research, because if you get these wrong, an analysis that could be done in a day might end up taking 3 weeks. That might make the project undoable. In addition to data handling and coding, sometimes you need to rely on parallelizing across multiple cores. Day 8 will cover the basics of these methods. And then day 9, the last day, is going to be wrapping up problem set 5 and exploring some of your research ideas that have come up over the course of the 2 weeks. So please, do talk to us about your research ideas. We're going to be keeping track of good things to bring up in discussion on day 9. And we're going to talk about what we think lies ahead in genomics research, what we think are some of the most promising directions going forward. Some of the key themes that are going to come up throughout the 2 weeks, I'll preview now. One is that empirical results are informative only if the analyses are well powered. So that's something you'll hear a lot, and saying that someone's analyses are underpowered is kind of a slur. So that's good. You should get into that mindset of insisting on high-powered research. Another theme is the 3 challenges of doing work with genomic data. One of them is the challenge of power, which is a problem because individual loci generally have very small effects. The problem of multiple hypothesis testing, which comes about because there are many, many loci that one could be studying. And the problem of population structure and stratification, which is the fact that individuals of different ethnic groups, or more generally, different genetic ancestries, have different patterns of gene frequencies and also correlation structures within their genome that can give rise to spurious associations with behavioral traits. So those will come up a lot, and ways of dealing with them. And a third key theme is that genetic effects probably often operate through environmental mechanisms. So this is something you'll see already in problem set 2. And the third problem on the Jenks and the Goldberger critiques, there's a common misconception that if something is an effect of a gene, that means it's biological. That's not correct. It could be environmental. And so understanding that in a deep way is actually an important goal of the course. Okay, so let me briefly say a little bit about the uses of genetic data in economics. And then the other speakers today will talk about the uses of genetic data in their own disciplines. So for those of you who are not economists, it might be useful to just say something about what economics is, especially because it wouldn't be crazy for you to think that, to be confused by the fact that a couple of economists are standing up here talking about genetics, and what does that have to do with economics? Economics today is not really defined very much by the subject matter of what economists are studying, but rather by the approach that economists are using to study whatever the topic is. And the basic approach in economics is that individuals are assumed to have goals, which are called preferences. The individuals are assumed to be pursuing those goals as best they can, which in econo-speak is maximization. Maximization of the preferences. And and that the individuals are facing constraints. And those constraints could take lots of forms. They could be legal prohibitions about what you're allowed to do. It could be, as it is in traditional economics, there's a limited budget. You want to buy lots of things, but you only have so much money, and you have to deal with the prices of things. Or it could be, as in behavioral economics, limits on cognitive ability or limits on willpower, given to temptation. And so in economics, any topic, whether it's the effect of sales taxes on cigarette taxes on smoking behavior, or whether people procrastinate in doing their homework. I mean, there are tons of things that are economics as long as you analyze them in this framework. The other thing which is, I think, distinguishing about economics is its goal, which is that much of economics has, at least as its ultimate goal, if not in the particular research question itself, to give prescriptive advice, which is called policy advice, and the way economists talk about it, to individuals, to firms, or to governments as a result of the analysis. Okay, so that's, and so why do we, as economists, get excited about genomics? Well, I think that there are at least 3 kinds of contributions that genomic data could make in economics. One is that it could help us understand some of the biological mechanisms that underlie behavior. So both in terms of the framework for economists, that would mean understanding the preferences themselves, where they come from, what they are, as well as potentially what some of the constraints are that people face, some of the biological constraints or psychological constraints that they face in making decisions. And that way, what we call genoeconomics is complementary with neuroeconomics. And neuroeconomics is about how the brain does decision making and one kind of set of biological inputs, and genoeconomics is about kind of genetic inputs. A second way in which genetic data can matter for economics is that it can play into empirical research that economists do already, that is currently done without any genetic data, but could be done better if we have access to genomic data. So there's kind of 3 broad types of examples that I have in mind. One of them is to use the genetic data as proxies for what economists call structural parameters of their model. So things like the basic preferences that individuals have, or the basic abilities that people have that affect what they're able to do, or things, internal production functions. So these would be things like the way in which your body translates the calories that you consume into how fat you get, or how much energy you have available. That would be an internal production function. Those are inputs to economic models, and they matter in the kinds of analyses economists do, but because we can't measure these things, or we have a limited set of ways of measuring these things right now, we often treat them as unobserved, or we have relatively poor measures of these things that we use in the analysis. And genomic data could be used to give us better measures. A concrete example of this is some work that David and Patrick and Mark and I are doing with Kevin Tom and Andrew Kaplan, which is on smoking. And we're looking at a life cycle model of smoking in the health and retirement study. So this is a study of how when people start smoking in their lives, when they might try to quit smoking in their lives, something that actually economists, there's a lot of work in economics on this. But what we're doing is incorporating some of the findings from the genetics of smoking, and in particular, genetic loci that are found to be associated with smoking that are involved in the nicotinic acetylcholine receptors, which are involved in the breakdown, the metabolism of nicotine when people smoke. And there's reason to think that, at least one pathway through which those genetic loci might matter for smoking is through affecting people's preferences for cigarettes. And there are other pathways that could matter as well. And in that project, what we're doing is studying the pathways through which these genetic loci matter, and also improving our power for studying life cycle smoking behavior, incorporating some of the genetic data in the health and retirement study. So that's actually something that's on the reading list for Kevin's lectures on structural models, and he may talk about that in his talk. Second example of using genetic data in non-genetic empirical work is for doing instrumental variables analysis. So that's the term economists use for Mendelian randomization, which Jonathan's going to talk about. So an example of that in the smoking context would be if we have genetic loci that we know matter for smoking through the metabolism of nicotine, we could use them to study the effect of smoking on something else. Like if you want to study the effect of smoking on the number of friends you have, well, there's all sorts of problems with just looking at running a regression of number of friends on smoking, all sorts of endogeneity problems and reverse causation problems. But if you believe that these genetic variants matter for smoking and don't directly affect the number of friends you have, then essentially by looking at the effect of the loci on number of friends, you know that that's going to work through smoking, so you can actually learn something about the causal effect of smoking on the number of friends. A third way in which this kind of work could matter in economics, and actually what I think is potentially one of the real big things, although very unsexy, is using the genomic data as a set of control variables. So this is on the reading list for this lecture. We describe how this would work formally in the supplementary section 8 of the Reitveld et al. 2013 paper. The basic idea is this. Suppose that you do a randomized experiment where you want to look at the effect of providing free preschool on educational achievement and maybe later life outcomes. So this is a very important policy question, but unfortunately it's a very expensive experiment to do because providing free preschool is very expensive. And so when this kind of experiment has been done, like with the Perry Preschool program, it's typically done in very small samples, maybe 50 kids or 100 kids, half of whom are assigned to a treatment group and half of whom are assigned to a control group. If we have some genetic data, well, actually, more generally, when you do this kind of randomized experiment, you can get an unbiased estimate of the effect of the intervention, whether or not you have control variables. Because by random assignment of people to treatment and control group, you're getting an estimate of the causal effect of free preschool. But if you include control variables that help you predict the outcome, then you improve your statistical power. And the reason is, what you do is you reduce some of the variation, some of the residual variation in the outcome, say educational achievement. You explain more of the variation in the outcome, and it's like you're clearing away some of the noise that otherwise you'd have to deal with when you're trying to see what the effect of providing free preschool is. And what happens in the regression is you end up shrinking the standard error on the estimate of the treatment effect of providing preschool. So genetic variables are not special in that regard. Any kind of control variable would help, but genetic variables, I believe, are going to be a very widespread source of these kinds of control variables. And in particular, if you have a polygenic score that helps predict educational attainment, that predicts a reasonable amount of the variance in the outcome, that would be a very good candidate for including as a control variable. And we'll have polygenic scores for all sorts of behavioral traits. You'll be able to control not just for the genetic predictor of educational attainment, but the genetic predictor for risk aversion and the genetic predictor for happiness, and you know, to throw all of these things into the regression. So I think that's gonna matter in many places. And then the final thing, the final potential payoff for economics is use in gene-environment interaction studies and studies of very closely related targeting interventions or differential effects of policies across people of different genotypes. So this is important across the social sciences, not just in economics. I know Dalton is going to talk about this as an important topic in sociology. Examples in economics include a paper by, or work in progress by Nicholas Papageorge and Kevin Tom, which is on the reading list. What they do is use a polygenic score for educational attainment to test theories of how people's ability and their resources interact in determining the amount of education that they get. So there's a lot of economics focused on that question. And they use the polygenic score for educational attainment as a measure of ability. And they find some very interesting things. They find that socioeconomic status and genes are substitutes when it comes to preventing high school dropout. So you can either have a high polygenic score for educational attainment or be high socioeconomic status in order to finish high school. But then in terms of completing college, socioeconomic status and ability are complements. So they find that you kind of need both high socioeconomic status and a high polygenic score in order to finish college. That's potentially pretty interesting in terms of the basic economic theory. Another example is on the reading list. It's work in progress by Sylvia Barcelos, Leandro Carvalho, and Patrick Turley. And it's using the polygenic score for educational attainment to look at heterogeneity in the effects of policy. In particular, they're looking at an increase in the compulsory schooling age in the UK on BMI. So there's a lot of work arguing there's a correlation between education and BMI, that more educated people tend to have lower BMI. There's a little bit of work using causal variation in educational attainment, which is one thing that they do. So they use the UK Biobank data and use an increase in the compulsory schooling age. So that's causal variation that then ends up mattering for people's BMI. It lowers their BMI years later. And they look at heterogeneity of that effect across different levels of genetic endowment, different predictors for BMI, and find big differences. So beyond this list, I expect that there will be other things, various creative, valuable uses of genomic data in economics that we can't anticipate now, but I suspect that some of you will be responsible for coming up with those. So that's, question.
Yeah, I have a question about the first example you gave.
Yeah.
So, left cost study, would you typically assume a genetic effect on the preference do not vary over left cost, or would you also try to look at kind of like a interaction between the aging process and the genetic effects?
So the question is whether you'd expect genetic effects on, say, preferences to be fixed over the life course, or whether they might vary with age. I mean, in terms of what I would expect, I think that would depend on what the particular genetic effect is. If it's an effect, that we might think is persistent over the life course, we might, so for example, if it's an effect on the way that the brain forms, then at least the effect on the brain is likely to persist throughout life. But how that ends up mattering for behavior could well change over the course of life, even if the direct biological effect of the gene is fixed. If, for example, to say an effect on preference for cigarettes, as people age, they're in different social environments with different pressures for smoking. They have different health risks that might lead them to be more likely to quit smoking at later ages. So even a fixed biological effect could end up mattering differently for the behavioral trait throughout life. So I think it's a very interesting set of empirical questions one could ask about that. Okay, so you guys didn't ask enough questions, which is why we're finishing early. So you need to do better for the next lectures, and I'll try to help and pepper the speakers with questions too. But we'll stop now and continue again in 20 minutes when Dalton's going to talk about using genetic data in sociology.